And we finished with a discussion of the spleen and the demise of the worn out erythrocytes. We're going to pick up the spleen again today, but we're going to twist the story slightly. But we're going to come back to the same idea of the peculiar nature of the endothelial cells that are forming the capillaries in the spleen. There are remarkably large gaps between adjacent endothelial cells in the splenic capillaries. And last time we emphasized the importance of those large spaces to permit the erythrocytes to leave the blood vascular compartment, enter the extracellular space of the spleen, where they could then encounter the macrophages. And if those erythrocytes had predominantly worn out or oxidized sugars on their surface, the macrophage, put your phone away, I'd appreciate it. The macrophage would then recognize that erythrocyte is being worn out and then basically attack it, gobble it up, endocytose it, and recycle the proteins. So what we'll do today is we'll bring up this idea of that red pulp where the erythrocytes are destroyed. But we're also going to bring up a neighboring region called the white pulp. And we'll bring this up because the white pulp is going to be kind of a germinal region for immune cells. So now within the spleen, there are going to be centers where we aggregate cells of the immune system. And those aggregated cells of the immune system now can sit there and be on the lookout for foreign proteins, for bacteria, even viruses that are within the blood, that have gotten out of the blood vascular compartment between the endothelial cells of the splenic capillaries and out into the extracellular space, the region, the so-called white pulp. These immune cells then can attack those foreign bacteria or foreign proteins. And this is going to be a theme through most of today's discussion. We'll start with the theme of the germinal regions in the spleen, where these germinal regions are watching out for pathogens in the blood vascular compartment. And then we're going to talk about the germinal regions in structures called lymph nodes. But before we can understand how the germinal regions in lymph nodes are functioning, we have to describe something known as the hemodynamics. Hemodynamics is a fancy term meaning that there is going to be a fluid movement from the blood vascular compartment into the extracellular space. And that fluid movement is going to result from the hydrostatic forces on the wall of the capillary. That hydrostatic force is going to be generated by the contraction of the left ventricle. Remember, if we follow the blood in the capillaries upstream, eventually we get back to the left ventricle, and the ventricular cardiomyocytes are contracting and generating a force on that fluid. That force is going to produce a hydrostatic force. That's going to be a force that's going to be against the wall of the capillary. We saw that the other day when we saw that graph of pressure versus the different kinds of vessels. So there's going to be a hydrostatic force moving the fluid between the narrow gaps separating the adjacent endothelial cells. Now, the gaps between the endothelial cells in the systemic capillaries are much, much smaller than the fenestrated endothelium in the spleen. But nonetheless, there are spaces. And fluid can move between those spaces. It can move through those spaces between the cells. But the spaces are so small that water moves out, but protein remains behind. Because the protein remains behind, then this means that there is an osmotic force, an osmotic draw, moving fluid back into the blood vascular compartment. So we're going to see two forces at play here. One, the hydrostatic force, which is a mechanical force, and then an osmotic force, which is basically a chemical force drawing the fluid back in. We're going to see that the hydrostatic force is greater than the osmotic force, giving rise to a net flow. After all, this is going to be a delta P, a differential in pressure. And we have a differential in pressure across resistance. We have a flow. Once the fluid now leaves the blood vascular compartment, goes out into the extracellular space, that fluid is now called lymph. And the lymph in the extracellular space is taken up by a series of lymphatic vessels. There are going to be lymphatic capillaries that drain into lymphatic ducts, and then lymphatic trunks, all the way back to the subclavian vein. So there's a kind of circuit. The fluid is leaving the blood vascular compartment, going out into the extracellular space, being taken back up into the capillaries and ducts of the lymphatic system and returned back to the blood. But through these tubes, the fluid, the lymph, is continually moving through these tubes. And as the fluid moves through the tubes, it will encounter these specialized regions called lymph nodes. The lymph nodes will be swellings, basically, openings in the lymphatic ducts and trunks. And as the fluid moves through these lymph nodes, within the lymph nodes themselves will be clusters called germinal regions where we have these immune cells. So again, we see that these immune cells are in a strategic location where they can be exposed to foreign proteins. Now, the foreign proteins in this case with the lymph are those extracellular spaces throughout the body except the brain. There's no lymphatic drainage out of the brain. But the rest of the tissues have a substantial movement of fluid through this lymphatic system. And then the fluid then moves over the cells and back to the blood vascular compartment through the series of tubes and through the lymph nodes. Then the third type of lymphatic tissue we want to talk about are the tonsils and Peyer's patches. These are probably the easiest to identify with because if you've ever had tonsillitis and you opened your mouth, you could see those swollen tonsils in the back of your throat. And so we know what these things look like. And basically what we're going to describe here is that the Peyer's patches are tonsils of the gut. And what they are is they are a section of epithelium. And the epithelium is going to be thrown into a series of folds called crypts. The crypts are now going to be sites where the bacteria are going to become lodged. And we now have specialized cells across the epithelium called M cells. And the M cells will actually gather proteins sloughed off from the bacteria, transport them across the epithelium, and then address these proteins to the germinal cells, the immune cells in the germinal regions right beneath the epithelium. Now, in this case, what we're doing is we're monitoring for pathogens that are coming in from the outside world, whether it's coming in through the air, whether it's coming in through water we drink, or whether it's coming through food we eat. We're basically monitoring for these pathogens coming into the body from the outside. And so we'll see then this commonality of clustering cells of the immune system to defend us from these foreign pathogens, whether it's in the spleen, whether it's in the lymph nodes, or whether it's just beneath the epithelium in the tonsils and Peyer's patches. Then the last type of lymphatic tissue we have to talk about doesn't do this. It's not important in itself of encountering foreign proteins and pathogens. The thymus plays a very different role. It's clearly a lymphoid tissue, but what the thymus does is it plays this role of positive and negative selection of T lymphocytes. We're going to see the T lymphocytes, whether they be T helper cells or T killer cells. These T lymphocytes play a really, really important role in the immune system. And the T killer cells particularly will seek out cells that have been infected with viruses and kill those cells. So these T lymphocytes, if they mistarget the protein and start to target proteins on our own cells, they can be lethal to us and we have an autoimmune disease. So one of the major things the thymus does is it encounters naive T lymphocytes that would have been born, naive T lymphocytes who have been born in the red bone marrow and moved to the thymus. And there's a kind of interaction here in the thymus between these naive T lymphocytes and stromal cells of the thymus. And what those stromal cells are doing is they're basically putting these naive T lymphocytes through a quiz, asking whether or not they can recognize protein, and if they do recognize protein, are they restricted to foreign proteins and not our own? So it's really important to properly select these T lymphocytes to be both useful, but also to not be dangerous. And we'll talk a little bit about that. And this time, we'll talk a little bit about adaptive and innate immunity. But let's start here with the spleen as a lymphoid tissue or organ. So I'll just do what I did yesterday and draw this sort of oval, call this the spleen. emphasize that there's a very heavy blood supply to the splenic artery. That the spleen's blood will be drained by the splenic vein. But that, as always, between the veins and the arteries will be the splenic capillaries. Now remember from yesterday that the splenic capillaries are unusual. So if we enlarge this and describe this as a splenic capillary, we find that there are gaps, sizable gaps, between adjacent endothelial cells. Now, in yesterday's story, the gaps between the adjacent endothelial cells played an important role because they allowed for the movement of the worn-out erythrocytes to leave the blood vascular compartment and cluster in a region known as the red pulp. Basically, the red pulp is an erythrocyte graveyard, because it's here that worn-out erythrocytes encounter macrophages. And the macrophages endocytose those worn-out erythrocytes. They recognize the oxidized sugars on the glycoproteins. They endocytose those erythrocytes, destroy them, and largely play a role in recycling the component parts. But there's another region here in the spleen I want to talk about. I'll circle this in purple. And this is called the white pulp. And another way of referring to the white pulp is that these are germinal regions for immune cells. So the body's immune cells are clustered in this white pulp, and these basically are leukocytes. But among these leukocytes will be T lymphocytes and B lymphocytes, will be macrophages, will be all manner of these cells of the immune system and dendritic cells. So the point of this whole discussion about the spleen is that because of the large gaps between adjacent endothelial cells that permit the movement of whole erythrocytes from the blood vascular compartment into the red pulp, you can similarly imagine how even smaller structures, such as bacteria, can leave the blood vascular compartment and now get out into the extracellular space. Because these bacteria, once in the white pulp, are going to be exposed to cells of the immune system, these regions of the spleen, particularly the white pulp, these provide a monitoring site. For blood-borne pathogens. Now, the fact of the matter is, if we have a terrible accident and we rupture our spleen, that's very dangerous. If you rupture your spleen, the surgeons need to go in there with a matter of 24 hours and get that spleen out, or you will hemorrhage and die. And so, but it turns out that you can remove your spleen and live a perfectly normal life. You have a slightly greater incidence of blood-borne pathogens, and it's worse if the surgery occurs in a child. But for the most part, many of the aspects of what goes on in a spleen can be taken over by the liver. But what I want to emphasize, though, is that we have regions within the spleen where immune cells will congregate And those immune cells now provide a site where we can monitor for blood-borne pathogens and mount an immune response to those blood-borne pathogens. So the general thinking here is spleen is a lymphoid tissue or organ that protects from blood-borne pathogens. Well, another place where we see this kind of monitoring for pathogens are the lymph nodes. And here I'll take this panel and say that the lymph nodes monitor for pathogens in the extracellular. You can read interstitial space. To understand how that works and how this can be, we have to start first with the ideas of lymph generation. And I'll take this portion of the panel and describe it as lymph generation and hemodynamics. Now that's a mouthful, but all we're going to be talking about here is how fluid moves from the lumen of the capillary out into the extracellular space. And we'll draw here, again, a linear series of endothelial cells. And we can label this then as a systemic capillary. Now, in contrast to the big gaps we find between endothelial cells of the spleen, which are sometimes known as a fenestrated endothelium, where fenestra is Latin for window, so they're big openings. These guys are pretty tight. These endothelial cells are pretty close to one another, so there are narrow gaps. Now, arbitrarily, we're going to talk about this capillary as having blood flow in this direction. Well, conversely, if we would then follow upstream, eventually we come back to the left ventricle. And remember, the left ventricle is the chamber surrounded by the myocardium. And when the myocardium contracts, it generates a force. And that force then places a pressure on the fluid in the ventricle. And as we saw in that figure the other day, those pressures then extend throughout the closed system, so that there are pressures in the aorta, there are pressures in the arteries and the arterioles. And although the pressures are dropping and dropping, we still have a substantial hydrostatic pressure of 20 or 30 or more millimeters of mercury. There is this hydrostatic, a kind of mechanical pressure. Much like the pressure when you blow up a balloon. When you blow up a balloon with air, you now have a mechanical pressure against the wall of the balloon that makes it expand. Well, there's a hydrostatic pressure here because of the pressure exerted on the blood in the closed compartments. So there's now a mechanical pressure outward. The gaps between the endothelial cells are so small that pretty much the only thing that's able to get through those narrow spaces is water and ions. But you remember from our initial discussion of blood is that something like 8% of the blood plasma was protein. Now, typically these plasma proteins have a molecular weight of around 40,000, so they're just too big to move through these tiny sieve holes between the adjacent endothelial cells. So what happens is we leave behind, as the water leaves, we leave behind these plasma proteins. So as the fluid is moving from this side to this side and water's being squeezed out, the plasma protein concentration increases. Because we've squeezed the water out, water's gone out into the extracellular environment, into the interstitial space here. Because the protein concentrations are increasing, we now have another force to reckon with, and that is the force called osmotic force. And the osmotic force is drawing or pulling the water back into the capillary. Water is simply moving from region of higher concentration to lower concentration, but it's moving now back into the blood capillary. Well, what we find when we look at these pressures, though, is that the hydrostatic pressure is greater than the osmotic pressure. Effectively, this is a pressure differential. It's a delta P. And you know, anytime you have a delta P, we have a resistance times the flow. So depending upon the resistance of the movement of fluid back into the blood vascular compartment, there will be a flow. But because the pressure, or the hydrostatic pressure, is greater than the osmotic pressure, the net movement of fluid is out into the extracellular space. So there's a net fluid movement into the interstitial space. Now, this term, interstitial space, is simply a fancy term. It means all of the extracellular environment around cells beyond the blood vascular compartment. Well, I think you can see, if this is all there were to it, if you had a hydrostatic pressure being greater than an osmotic pressure, and you had a net movement of fluid from the blood vascular compartment into the extracellular space, that you'd collect fluid out into the extracellular space. So you need to have a drain. You need to have some kind of a return system. And so what we find is out here in the extracellular environment, in that interstitial space, will be these structures. They have little valves on them. They're in fact lined with an endothelium. But these are termed the lymphatic capillaries. These tiny lymphatic capillaries that are not really much bigger than the blood capillaries will drain into larger structures called lymphatic ducts. The ducts then drain into trunks, and then the trunks eventually then will carry this fluid back to the subclavian vein and back to the blood vascular compartment. So it's a kind of circuit here, it's a kind of cycle. Fluid leaves the blood vascular compartment in the capillaries. That fluid then is taken up by the lymphatic capillaries, and then this fluid is then taken back to the blood vascular compartment through a series of tubes. Unlike the cardiovascular compartment, where there's an intrinsic pump, the heart is pumping fluid through those tubes. There's no intrinsic pump here in the capillaries or trunks or ducts of the lymphatic system. This is all driven simply by mechanical movement. As you exercise, as you move around, your skeletal muscle will in fact put pressure on these structures and move fluid through the ducts. And you know this intuitively. If you've ever been in bed for a couple of days and you haven't been moving around, one of the things will happen is your hands and your feet will start to swell. And they start to swell because of this accumulation of fluid out here in the extracellular space, and you're just not bringing it back into the lymphatic system to bring it back to the subclavian vein as fast as it's being produced. So it pools out in this extracellular space, particularly in your hands and feet. But what I want to emphasize is that at intervals along the return system, along the tubes that are returning fluid back to the subclavian vein, we're going to find these structures that look a little like kidneys. And this is going to be a lymph node. And there will be thousands upon thousands of these along the ducts and trunks leading back to the cardiovascular system. And there is a flow of fluid here into the lymph node. The flow of fluid comes in through the so-called afferent lymphatic vessels. And it leaves here through a series of vessels known as the efferent lymphatic vessels. It's not too much of a stretch to imagine the lymph node as a kind of lake, and imagining the afferent vessels as being a series of streams leading into the lake, and the efferent vessels being a series of streams that are draining the lake. But you know that you can have fairly fast movement of the fluid in these streams. And once it reaches the large three-dimensional diameter, three-dimensional structure of the lake, the fluid rate flow slows down. But within the lymph node, there are these structures again, and I'll show them as circles, known as the germinal regions. And these germinal regions are really collections of cells of the immune system. So now look what we've done. Throughout our bodies, we have these capillary beds. And fluid is moving out of the capillaries into the extracellular space. And then once in the extracellular space, it's being taken up by the lymphatic system and moved through these series of tubes. So throughout our bodies, this fluid is going to bathe all of our cells. And any pathogens that may be out here in the extracellular space will be swept in the flow into these lymphatic capillaries, ducts, and trunks. And in the process of flowing through the lymphatic system, those pathogens, those bacteria and viruses now, will flow through this system of lymph nodes, flow through and around the germinal regions, and these germinal regions will now contain cells of the immune system, which are strategically located now to recognize the foreign proteins moving through this system. So as again, just as we saw above where we were monitoring for bloodborne pathogens, we can now use the lymph nodes to monitor for pathogens throughout the body, any place where there's extracellular space. Perhaps the best example of where we see these lymph nodes as becoming highly responsive, too responsive, is something called the bubonic plague. And the bubonic plague is a case where there's a bacterium, Yersinia pestis. And Yersinia pestis gets into the system in some way, and often through a flea bite, and then will make its way here into the lymph nodes. And the Yersinia pestis bacterium can take up housekeeping within those lymph nodes and start to divide. And the term bubonic plague comes from the swellings of the lymph nodes. They didn't know in 1340 that they were in fact lymph nodes. They just knew that they could get these these swellings, usually in the inguinal areas and the axillary areas, and these lymph nodes would start to swell. And then they would turn black, and that was the bubonic or Black Plague. And it was lethal. It killed about a third of the population of Europe. And probably, although it's not as well chronicled, it looks as though there was a similar movement of the plague that came out of the Black Sea area. The Black Sea area is a great trading region. And so it moved to the west into Europe, but almost simultaneously, it's thought to have moved east into China. And we think that probably about 25% of the population of China was killed at that time too, simply as the plague swept around the world. And this was all a bacterium that became lodged in the lymph nodes and produced those buboes. And the bacterium still exists. It turns out they've actually done the genetics on it. A couple of years ago, in the process of digging a new tube in London, they came across an old cemetery, and they dated the cemetery and realized that it was put there specifically at the time of the plagues. And so they took, and I don't know why they took 2 samples, but they took 2 samples, and they took a little bit of pulp out of the tooth, and they sequenced the DNA from the Yersinia pestis bacterium. They found Yersinia pestis bacteria in these people's bones and teeth. They then sequenced the DNA and found it looks like it's just about the same Yersinia pestis bacterium we see today. In fact, the Yersinia pestis bacteria we see today is commonly found around here in the prairie dog communities. So if your dog goes out and gets in touch with some of those prairie dogs, it might get sick, so watch out. But the point there was the whole concept of the fluid moving through those lymph nodes. Now we're going to change gears a little bit. And we're going to talk here about the epithelial associated lymphatic system. Now, that's a long-winded way of saying tonsils and Peyer's patches. But what we're going to see is we have a series of germinal regions, a series of clustering of immune cells located right beneath the epithelium of the pharynx, and then at intervals all along the GI tract. The function of these tonsils and Peyer's patches, these epithelial associated immune sites, is to monitor for pathogens that are coming into our bodies from the outside world. So we'll take this panel and label it as tonsils and Peyer's patches. As lymphoid tissues, organs that monitor for pathogens. In the outside world. Probably the easiest one to think about are the tonsils. The tonsils are located in our pharynx, and sometimes if you have a sore throat and you go up to a mirror, open your mouth, and look in the back of your throat, you'll actually see your tonsils. Actually see them as these little bumps that are inflamed and sort of red. So the tonsils are in the pharynx, but the Peyer's patches are similar structures along the GI tract. Now, I'm going to draw sort of a cartoon here of one of these tonsils or Peyer's patches. And initially, it's going to look like a kind of exocrine gland, but it's not. What we find in the regions of the tonsils and the Peyer's patches is that the surface epithelium is thrown into these folds. And for the tonsils and Peyer's patches, these folds have a name. They're called crypts. And using the crypts, we play a pretty dangerous game. What we do is we actually will capture bacteria and lodge them in the crypts. And of course, because this is an epithelium, it sits on a basal lamina. So the apical surface of these epithelial cells are facing the bacteria. Then down here on the basilar side will again be germinal regions. So again, these are clusters of cells of the immune system. Then there's an additional cell type here, and I'm going to put this one in as orange. It's a modified epithelial cell. It has a name called an M cell. And what the M cell does is it transports proteins across the epithelium. So these bacteria sitting in the crypts, stuck in the crypts, are sloughing off proteins. The proteins bind probably to proteins on the surface of these M cells. They're endocytosed into vesicles, delivered then to the basolateral site, and these foreign proteins then are dumped out into the extracellular space. But the extracellular space here is right adjacent to the germinal regions. So this is a way now of presenting cells of the immune system with these pathogenic proteins from the bacteria. It's a risky thing to do, because if you don't mount an immune response to these pathogenic bacteria before they overwhelm the system, you're in some real trouble. But we take the gamble, because what we'll do is we'll generate specific immune responses to these bacteria and then become immune to these bacteria for the rest of our lives. Now, sometimes it doesn't work out so well. And among the population, there are some people who will end up getting a chronic infection of these bacteria. Usually a streptococcal or staphylococcal bacterium will take up housekeeping in these crypts. And the immune system never quite gets it together to get rid of these bacteria. And then something happens and the person becomes stressed, they're slightly immunosuppressed, and these bacteria start to divide and multiply, and the person ends up with tonsillitis. Now, the typical medical procedures these days are that if you have recurrent bouts of tonsillitis, they'll go in and surgically remove the tonsils. They'll do a tonsillectomy. But we now recognize that these tonsils are an important part of our immune system, and that maybe we shouldn't be chopping them out willy-nilly. But that wasn't true so long ago. If you talk to people in my generation, probably 80% of them, just routinely, when they reached 7 years of age, went into the doctor's office and said, "You need a tonsillectomy," and they chopped out the tonsils. Now, I suspect in this group, no more than a third of the people have had tonsillectomies. How many of you had tonsillectomies? Yeah, maybe about a third of you. And those are usually people who have had recurrent bouts of tonsillitis. And so then that relieves these individuals of probably the underlying bacteria and these recurrent bouts of strep throat. But for most of us, we can actually keep our tonsils throughout our lives, and they will be useful in terms of mounting an immune response. But the major point I want to make here to bring the stories together is where with the spleen we were monitoring bloodborne pathogens, and with the lymph nodes we were monitoring pathogens in the extracellular space. Here we're actually monitoring for pathogens coming in from the outside world, because these tonsils and the pharynx are really strategically located. So every time you take a breath, every time you take a bite of food, every time you take a drink of water, these are strategically located to gather some bacteria and expose them to your immune system. So we can now monitor for pathogens in the outside world through this system. So any questions to there? Yes?
If you don't have your tonsils, can you still get strep throat?
If you don't have your tonsils, I hate to say no, but you shouldn't be able to get a strep throat without your tonsils because you have no site for these bacteria to take up housekeeping and then start replicating themselves. But those of you who had tonsillitis will know better than I. So, has it cured your strep throat? It has? Okay. Okay, so much for that theory. All right. So, I don't know. Can't explain it. Right? But I think it's at least helped some. So what I want to do now is I want to go on to another lymphoid tissue. The lymphoid tissue that I want to emphasize here is something known as the thymus. And the thymus doesn't play this role in itself for monitoring for pathogens. It's a selection site. It's a site where we select for T lymphocytes. Now, as we work our way through the immune system tomorrow and Monday, we'll find that T lymphocytes play a really important role in what is known as the specific immune system. But the T in T lymphocytes actually stands for thymus, because all T lymphocytes, as part of their life cycle, have to pass through the thymus. And the thymus is the place where we, in fact, select for the efficacy of the naive T lymphocytes. So the thymus is a sizable organ, particularly when we're young. As we get beyond our adolescent years, that it begins to atrophy. But for the most part, when we're young, it's really busy trying to mount an immune response. And it's located right behind the sternum and right in front of the heart. So it's sort of in your thoracic area. But the thymus, let's talk about the thymus as lymphatic tissue organ that selects for functional T lymphocytes. So you remember that all cells of the immune system, all blood cells, originate from the red bone marrow. So we find that naive T lymphocytes leave the red marrow. Their destination is the thymus. And these naive T lymphocytes will enter the thymus. So I'll put these in as circles. These are intended to be naive T lymphocytes, fresh out of the red bone marrow. Once they get to the thymus, we name them differently. We call them thymocytes. But within the thymus, there are specialized cells, and I'll put these in orange. These specialized cells are called stromal cells. They appear to be modified epithelial cells. And it's these cells that select for functional T lymphocytes. Now, I'm going to anthropomorphize this a little bit because I think it's just easier to understand. What happens is these naive T lymphocytes, fresh out of the red bone marrow, come into the thymus, and they sidle up to the stromal cell. And there's a kind of handshake. Hello, how are you? Some small talk. And they greet one another, and the stromal cell says to the naive T cell, it says, can you recognize proteins? Do you have transmembrane proteins on your surface that can bind to other proteins. And if the naive T lymphocyte says, no, I don't have any of those proteins on my surface, I don't have any transmembrane proteins that can recognize other proteins at all, then the stromal cell looks at the naive T cell and says, you're useless, go die. And in fact, what happens is the stromal cell then withholds growth factors. It doesn't secrete growth factors to allow this cell to survive. The growth factors are important in turning off the suicide machinery, the apoptotic machinery inside these cells. So without those growth factors, this cell will basically turn on its suicide machinery and the cell will die. But then another cell comes along and comes up to the stromal cell. There's the handshake, hello, how are you? And the stromal cell says to the naive T lymphocyte, says, can you recognize protein? And do you have proteins on your surface? Do you have transmembrane proteins that recognize other proteins? And the naive T cell says, sure, I've got lots of them. I can recognize any number of different proteins. And then the stromal cell says, well, can you recognize all proteins, or can you recognize only proteins that are from our own body? And the T lymphocyte says, I can recognize any protein. And the stromal cell says, you're dangerous, go die. And withholds growth factors so the cell doesn't survive. And it's true, because if this is going to be a T killer cell and it grows up to be a T killer cell that recognizes proteins on your very own cells, these T killer cells will then go out and kill those cells. That's a classic form of an autoimmune disease. So you don't want to have proteins that recognize their own proteins. And so the ideal situation here is that the naive T cell arrives at the stromal cell, and there's an interaction. And the stromal cell recognizes that the T lymphocyte has transmembrane proteins that can recognize other proteins. We'll see these are MHC1 proteins primarily. And since this has those kinds of proteins, and then it's asked the question, can you recognize our own proteins? And there's a whole cascade of events in here where they check out all manner of different proteins from the body. And if there's any time where this T lymphocyte can recognize a protein on a stromal cell, then the stromal cell withholds growth factor and the cell dies. But if the naive T cell recognizes protein, but also only recognizes proteins that are foreign, that is, they're not contained within our own component of cell proteins, then this is a safe T cell. And then that T cell will be released out into the outside world and will then go to a germinal region and sit there and wait to find some pathogen that it can recognize with something called its T cell receptor that we'll talk about a little later. So what this cell is doing is it's going through a process of so-called positive selection. And the positive selection is simply a fancy way of saying it recognizes the thymocyte. The naive T cell recognizes recognizes protein. But simultaneously, it's going through negative selection. And the negative selection is selecting thymocytes that recognize only non-self proteins. So this guy plays a really, really important role in selection of these T cells so that we don't generate some form of autoimmunity. In the last couple of minutes, let me lay out where we're headed. Do this in the backboard. When we talk about immunity, we divide the immune system into these two great categories. One is said to be the innate or non-specific. The other is said to be the adaptive or specific. The adaptive branch includes B lymphocytes, T lymphocytes, and a third cell type called dendritic cells. The innate or non-specific branch includes cells such as macrophages and cells called granulocytes. The granulocytes come as neutrophils, eosinophils, and basophils. And those fundamentally are the cells of the immune system. These, when I blithely talk about those germinal regions containing cells of the immune system, that's this set. And what we'll do next time is we'll talk about the difference between the innate and adaptive immune systems.